Today we're talking about the second part of protein synthesis, translation. The first part is transcription, and the second part is translation. So let's get started. Here we go. So let's just go back in time for a second and remember some vocab terms. Remember that ribosomes are either found free-floating in the cytoplasm or attached to the rough ER. Also, back in time, if we look at a protein, remember that the polymer of a protein is a protein, and the monomer of a protein is an amino acid. And so an amino acid is nothing more, a protein is nothing more than a series or a string of amino acids, just like we have here. And then the string of amino acids gets folded up into these cool structures over here, and then it's a protein, and it does a job, or it plays a role in structure in your body. So moving right along. What is translation? Translation is the moving of genetic information from RNA to protein. We call this translation. When we're looking at RNA and DNA, we're talking in the language of A's, T's, C's, and G's, and U's in the case of RNA. When we go to protein, we're speaking a new language. We're speaking in the language of amino acids. So we're translating into a different language. Whereas DNA to RNA, that was transcribing. We were changing the message. We were moving the message from DNA to RNA, but we were still speaking the same language of A's, T's, C's, G's, and U's. Let me go back. So mRNA, or messenger RNA, as we learned about in transcription, is read 3 nucleotides at a time. If you look at this picture here, you can see right here, AUG, that is a codon, 3 nucleotides. So these 3 nucleotides, they specify a specific amino acid. So codon is a group of 3 nucleotides that specify a particular amino acid. Let's talk a little bit about amino acids then. Each codon is specific to an amino acid. There are 3 nucleotides in a codon. There are 4 possible nucleotides to choose from. So there's 3 nucleotides, and there are 4 possible nucleotides. So therefore, you have 64 possible sequences. And so that means that there are 64 possible UUA, UUG, UCU, UUU, AAA, 64 of those. However, there are only 20 amino acids. So here are our amino acids. This is a little bit small, but this is the codon wheel that you'll get in class. And so the amino acids tend to end in a sound like ene, like threonine, asparagine, lysine. There are 20 of these, but there are 64 ways to get there. There are 64 codons. And so as you can see here, multiple codons, like CUU, CUC, CUA, those all code for leucine. So let's try a few of these. If the mRNA strand has a codon that reads UAA, which amino acid does it code for? So we're going to start in the middle where the letters are biggest, and it's UAA, which is stop. It's a stop codon. When a ribosome is reading an mRNA strand, it's going to stop and release the protein when it's done, when it reaches the stop codon. Let's try another one. If the mRNA reads AAA, we're going to start in the middle here, AAA, that codes for lysine. It could code for, let me find it, methionine right here, which is AUG. AUG also methionine, but is also the start codon. So the ribosome is going to clamp on to the mRNA strand, and when it reads AUG, it's going to start making the protein. It's going to keep reading and keep reading the nucleotides, or the codons, until it reaches the stop codon, and then it's going to release. So how is mRNA read? Here is our mRNA sequence. It's a very short one, but you can see right here, here's our start codon, AUG, and it's going to read, here's a codon, here's a codon, here's a codon, and it codes for, or specifies, these amino acids until it gets to stop, and then the ribosome is going to stop making the protein. So if you can picture it, the mRNA strand is made in the nucleus. The RNA polymerase reads the DNA and spits out the mRNA, and that all happens in the nucleus because the DNA is safe in the nucleus. The mRNA leaves the nucleus, goes into the cytoplasm, finds a ribosome, and there protein synthesis happens. The ribosome assembles around the mRNA strand. So you can see here there's the larger subunit and the smaller subunit of the ribosome, and it sort of just clamps on around the mRNA strand. This is the mRNA strand, and it's reading, as you can see here, UUU, that's one codon, and UCG, that's another codon. And you can see that there's something else happening here. This right here and this right here, these are called tRNAs, transfer RNAs. They transfer the amino acids. So this sequence of amino acids didn't come from nowhere. It had to come from somewhere. And so the tRNAs, as you can see, this one is coming in, and it's going to read the mRNA. It's going to be matched to the mRNA right here by this. This is called an anticodon, and it's going to deliver the correct amino acid. So amino acids are right here on the tRNA. They're kind of at the top. So this is generally what we look at, and so the amino acid is going to be right here. This bottom part is called an anticodon. It's a complement to the mRNA, which is confusing, but we'll explain it further. Let's practice this. So tRNA has an amino acid on the top of the molecule and an anticodon on the bottom. And an anticodon is a complement to the codon to match the amino acid to a specific codon. So you can see here's the tRNA, and this is the mRNA. Here's the codon. This is the anticodon. This is part of the tRNA. And so when the mRNA reads CGA, it knows that it needs to bring an arginine, and that is added to the chain. So let's draw this out here. Let me see if I get my pen working. If we were to draw an amino acid, it sort of looks like this. Here we go. And then, so this is, this is a tRNA, and here's our amino acid. And down here we're going to have the anticodon. It's a pretty poor drawing. And so we're looking for the codon, the anticodon for serine. So let's just put a little S in here. This is our amino acid serine. And we're going to draw an mRNA strand. And to code for serine, we have UCU. There's a whole number of them, but let's just use UCU. We have UCU. If the mRNA reads UCU, actually it'll keep reading in this direction, so let's do this so that's not confusing. It's going to be reading in this direction. It's going to read UCU. The appropriate tRNA is going to be AGA. That's what's going to be on the anticodon. Let's try another one. Oops. Valine. Actually, let's move on to the one after that. Methionine. Let me go to my pen. Pen should be working. Here we go. So if we have our tRNA that looks sort of like this, here is our methionine, and here is our anticodon. Here is our mRNA, and we're reading in this direction. If we're looking for methionine, the codon is AUG. So down here, the codon, the mRNA is going to read AUG. The tRNA is going to read U. I'm going to write it over here so you can see it. UAC. That will be the anticodon on the amino acid. All right. So the ribosome reads the RNA one codon at a time, right here. So it's reading AAA, GAU. It's matching, or it's finding the appropriate tRNA with the anticodon with a specific amino acid. And then it strings these amino acids together. These string of amino acids are shipped to the Golgi apparatus to be folded, and then the folded, strung together amino acids are now a protein, and they can provide structure to your body, or they can do jobs in your body. There is a video on Moodle. It is translation, the computer animation. I recommend that you watch that along with these notes. And the same is true with transcription. Please feel free to come and ask me any questions.